We all get so excited once we get the university approval. We hurry to start lab work without any planning. After a few weeks, we discover we should have prepared enough material for the analysis. We get back and prepare more. Then we discover we have different results. We get stuck for a few weeks or months. Then we decide to change the point after a few trials. This time we decide to plan our work. Before you start the lab work, you need to plan your work. Split your project into parts and start with the first part during planning for successive parts. That will give you a head start in discovering any problems going with your results. If you are a chemist or pharmacist, you need to know you will never prepare the same exact compound twice. Start by preparing small quantities. Test them. You may need mass spectrum or NMR results for verifying your compound. Once you are sure of the method that you used in preparing the target compound, prepare large amount that is enough for all analysis and applications. Use a single batch for the whole work, gives you consistent results. Start collecting data in systematic method. Stop after few results and make sense of your results. It may sound very silly and outdated, but write everything with dates in a note. Trust me, sometimes when you have a problem, you will go back to all dates. For example, if you get a contamination, you can just look at the samples and exclude all the results before the date where you got the contamination. Number your vials and the samples with as much identifying information as possible. Don't use numbers only. If one of the numbers is deleted for any reason, you will end up with the wrong vial. Use dates, numbers, and initials. Rewrite all your week work on your computer and back up your data once a month on the cloud or a drive. Or even you can use the cloud for recording all your results if it is secured. Write every single detail from your note. Try to organize information as possible. At this point, don't try to write anything except your lab work and if there are any observations. Remember to record your samples with clear dates in a table. This will make your work easy to track. Read well during your first stage of research. A lot of students don't read during the first stage of their lab work, only at the last stage during writing, which is completely wrong. Read about your study, who did similar work, and what were their results. If you start with a pre-knowledge of what is expected to happen, you are likely to discover any error in collecting your data. But be careful of bias during a research stage. Pre-anticipating the results can make you overlook a new discovery. Again, your reading will make it easier to understand the trend of your results. You will be able to interpret them easily. Write in your notes the name of the paper that matches your results, which makes it easier when you start writing to get back to your note and refer to that work. Divide your work into parts. Once you finish a part, start writing the results and interpret them. Don't keep working inside the lab without trying to write your results. You don't have to write your introduction or start writing your thesis. Start by writing part by part. You will find it that you can even publish each part separately and have a thesis by publication. So what if you hit the wall with your results? What I mean, if you prepared a compound that you hoped that it would show antimicrobial or antifungal activities and your compound failed to pass those tests, it is not the end of the world. Think of it that way. Now at least everyone knows that this compound is not good for this purpose. State suggestions to improve the results in the discussion section. Remember, if it is bad, at least someone proved it. It is like telling others, don't try this recipe. I already tried it and it failed. So what if you need to repeat one last analysis and there is no compound left? Yes, it does happen, and a lot. The scenario goes like that. You submit your paper and you get feedback about certain analyses that you should include for the publisher to publish your work. You go back to the lab, prepare your compound without testing the compound. You go straight to get the analysis the publisher asked for. The results come back with odd or unjustified results. It may sound weird, but it happens a lot with chemists and pharmacists. Before running to the lab, justify the reason for not performing this analysis by saying you used another method that gives similar information. If they insist on including this analysis, prepare your compound and repeat 1 or 2 of your analyses to make sure you prepared the same compound. Sometimes your luck runs out. You try to prepare the compound, but you fail. The answer to that may be better, but you need to consider sending your work to another journal. This is why I advise you to prepare enough amount of your compound and keep it in a safe place in case you need it at any stage of your research. What if some unexpected complications happen? In one of the clinical trials in the USA, some data were lost during the hurricane. The discontinuation of the trial was reported when the study was published, and the reason for the discontinuation. Remember, there is no reason to repeat the whole trial because of a reason that can happen again. The next session will be about the writing stage during the research and after collecting all data.